The overall goal of this procedure is to use immunohistochemical staining techniques to evaluate the expression of B7-H1, PD-L1 on paraffin sections of human pancreatic adenocarcinoma tissue. This is accomplished by first removing the paraffin from and rehydrating the sliced tissue sections. The second step of the procedure is antigen retrieval, which involves the use of a pressure cooker. To stain the tissue, an antibody staining procedure is followed by signal amplification and DAB color development steps. The final steps are to dehydrate and mount the tissue. Once complete, microscopy and image analysis can be used to visualize membranous and cytoplasmic B7-H1.
This method can help answer key questions in the study of pancreatic adenocarcinoma by giving us a better understanding of how B7-H1, PD-L1 contributes to the suppression of anti-tumor immunity in the tumor's microenvironment.
The main advantage of this technique over existing methods, such as staining on cryosections, is that it has been optimized for use with paraffin-embedded tissue sections, which are often more easily available and more readily stored.
Reliable antibody staining on slides isn't possible without removing the paraffin first. So to prepare slides with paraffin-embedded tissues, first, bake the slides at 55 degrees Celsius for 20 minutes. Next, in a